Hi everyone, come back to the topic related to the epigenetic. One of the epigenetic data that can be come from the ChIP, which means chromatin immunoprecipitation, which means on the DNA fragment they may not only the nucleotide itself, rather than they may have the protein that binding to the DNA region. And actually, if we can know exactly on a long DNA sequence, we can know where the special protein bind into the DNA. It may help us to explain about how a gene is regulated, and because of that we can understand more about, for example, the cancer, how it progress. And right here, back to the question, like how can we know exactly this special protein bind into which region of the DNA sequence? Very simple, just like we fragment the DNA, and the next step you can see like the protein may be bind into the specific DNA fragment but not on. And the next step is just like we design the antibody that will bind specifically into this DNA fragment. And the next step just like we wait for the connection between them, and if the DNA fragment it doesn't have the protein, it will not be keep in this one. And the next step is washing, which means it will remove all of the DNA that doesn't have the specific protein. And the next step is just like we collect them and we sequencing just like the normal sequencing step. And based on that, if we collect this rich information and we align to the reference genome, we can know where these rich are aligned. And because of that, it can have to answer the question related to how a gene is regulated. For example, right here, if we compare between the normal and the colorectal cancer, between here's the region of two genes, TAT85 and TLR3, and right here just like three track, it's just like three different type of protein, different type of chipstick. And if we can see a simply idea that between the normal versus the colorectal cancer, there's going to be the difference between them, just like on the specific region, on the same genomic region right here. But on the normal, they have a higher level, higher protein binding into this region, while the colorectal cancer it doesn't have. And similarly, a few region that may have like more specific of this protein bind into the DNA region, it may cause something. And right here I just want to explain a little bit about how it can change the gene expression level. As you can see right here in the 3D structures, actually the gene is not like a linear model. Actually it's just like in the 3D space it may work like that, and very far away from the transcription site, where in the remote region you can see a DNA region right here. It contains and it fall in the 3D structure. As a result, it can extract the transcription factor, and also when it fall, it also form with the promoter as a hub model. And right here, it can extract, for example, like the RNA, just like enhancer RNA. And together, if the enhancer is very strong, right here on the second structure, it's just like a very light region, and also like a higher signal of the protein. Actually, for the specific, the enhancer region, it usually marked with these markers just like the H3K27 acetine. And similarly for another protein track, and actually this track, if together on a specific DNA region, they occur together, it's usually the active enhancer region. And because of that, the super enhancer region over here, it can make a gene product, for example, like it's the same gene, but if the relative enhancer region they are very strong, it can increase the number of product. For example, you can see right here, normally it may have like only 2 transcription products, but right here they may have 5, which means like it increase a lot of the product of the transcription. As a result, we can see like understand about the epigenetic that can help us to modelize the how a gene is regulated